Hello, everyone. I'd like to welcome our next speaker, Bartek Wilczynski, who flew all the way from Europe. He will be talking about understanding your data with Bayesian networks. And he has been working on high-throughput data analysis ever since his work in the National Lawrence Livermore Lab, working on DNA sequences analysis. And since 2008, he has been involved in the development of BNFinder, a Python software for finding the structure of Bayesian networks for machine learning purposes. He holds a PhD in mathematics and is the assistant professor of computer science in the University of Warsaw. So let's welcome Bartek.
Thank you. Hello. Can you hear me? Yeah. Okay, good. Thank you very much. It's been a nice meeting, although I have to say that it's a little bit outside of my natural habitat, which is mostly either at the university or at a scientific meeting. So I actually have to say that I very much enjoy, and I've already learned many things about current versions of software. It seems the more I am teaching programming to students or statistical data analysis, the more I'm depending on older versions of packages, and it's kind of cool to see those kind of bleeding edge live demos of things that have been written very recently. So what I'm going to talk to you about today is actually, so I've changed the title of the talk a little bit, and I've changed the slides even more after I've heard a few talks. So I'd like to start with a little bit of a, I understand that not everybody is familiar with Bayesian networks, so I'll give you intro into basically what is this all about. So to start you off, I was wondering what to start with, and naturally XKCD is something that is a very nice thing. It always has a comic about something I need to talk, and one thing is that with Bayesian statistics, there's a lot to do with causation and correlation. And causation is a difficult concept, and this is why I wanted to start you off with thinking about confusion. So to get you a little bit more confused than you are now, I'd like to maybe bring a bit of information about teaching. So this is, it's always, when you think about teaching, about conveying difficult concepts to people who are not necessarily very familiar or very much into this. You may wonder how to actually best do it, and Professor Mazur in Harvard has been teaching physics, which is also many students find difficult, and he was studying what are the different things that make students learn better or worse, and he was asking them questions, showing them a experiment, asking them in a questionnaire different things, and one thing he found is that confusion seemed to be helping. So just let's look at the data. It's not too much of data. It's actually 8 numbers, but it's misleading actually. If you know that the first and second column sum up to 1, you'll actually notice there's only 4 numbers, but it's easy to see that for 2 different experiments, when he asked students whether they were confused while he explained the experiment to them, and then asked them some question about the experiment, those who were confused tend to, it seems that the questions weren't very easy, because either way, majority got the answer wrong, but those that were confused were actually answering slightly better than those that were not. So you think, what is the model? How do I understand this data? What is it telling me? And one very simple model, so the model on the left is basically the simpler version. So it tells you if a student is confused, then he or she is getting the answer. So being confused itself must be helping in understanding. Or maybe there's an alternative explanation, so maybe we're not, with our data frame or our data set is not telling us everything. Maybe, and that's my favorite explanation of this particular finding, is that maybe there's some hidden variable, like for example, whether a student was paying attention or not. So for example, if you look at the right-hand side, there's this model which basically includes this additional variable, which is a student paying attention or not, and naturally, to be confused, you need to pay attention. If you don't pay attention, you're just watching the birds out the window, then most likely you're not being confused by the explanation. And then if you're not paying attention, you're also less likely, although, well, this depends also on the speaker skills, but you're then also less likely to give a correct answer. And then these are just cartoon models, but they're helpful. So you, like, one key thing is the intuition is here that we will think about variables, so things that describe our observations. We describe those observations in variables, and then this kind of act of connecting them with edges is somehow intuitively corresponding to what's causing what, although it's a very high level now. And we're just drawing these two cartoons. It's very hard to say whether one is true or the other is true. I think actually confusion is helping people think about things and basically make them make an effort, which is why I'm trying to basically start you off with something completely irrelevant for the rest of the talk. But basically, what I'll try to tell you in the rest of the talk is how to basically move from kind of drawing cartoons and having intuitive understanding of concepts such as causation to more quantitative models such as Bayesian networks. So what do I mean by data? I'm actually coming from a computational biology field, but I'll show you some examples of the applications that we have been working on later. But for the purpose of making this sort of common ground, you can imagine just a questionnaire that a doctor is giving to his patients in a hospital or in a private practice, and there's some data that we have about different people. It's kind of, I have a pointer, I would probably could use it, but it's kind of tricky to show on both screens at the same time. We're still waiting for this technology that will allow us to point to one and then give you kind of a mirror image of the pointer on the other one. But basically, each row corresponds to a patient or a person, and then each column corresponds to some piece of data about this person or a variable. There could be missing data, which I won't go into now, but it's also possible to handle missing data with these techniques that I tell you about. So basically, we have this kind of tabular data. It's a kind of very, very common. I don't think that I should spend too much time on that. And now, what we want to have is some sort of a network of connections. And this is, again, at this intuitive level where I have those arrows just pointing, like saying, for example, that with increasing age, the chance of having heart problems is increasing. In general, statistically speaking, that's something that we kind of see, but it's also, for example, having a stressful job or would also have some influence on the same variable. So in principle, one thing that I want you to think a little bit about is that the arrows here are kind of intuitively, I think there's not much, people don't have an issue with the fact that we have those arrows with a, so there's basically an orientation in this influence, but most of those arrows, if you think long enough about them, they're not as easy to draw as you think. So for example, let's take this kind of stressful job leading to heart problems. So it's actually shown scientifically that having extensive exposure to stress can cause you problems. But then once you have those problems, you're more likely to change your job to something less stressful, right? And if you actually look at the statistical data of many patients, if you're looking at someone in an advanced age, for example, with heart problems, then actually the chance that they will still be doing their stressful job are less likely. So usually, if you look deeply in your data, then there will be also some additional, maybe unexpected connections that might make things more difficult for a data analyst who's trying to look at those and find them out from the data. So kind of, sorry for this slide, I It's a bit text heavy, but basically there's a few things that you need to know about Bayesian networks. So this previous slide was kind of a very, very abstract and intuitive, hopefully, but basically we're talking about a mathematical object. So what we have for a Bayesian network, it's a graph like the one I showed you. It's just one assumption that's quite technical, and it happens to be important, is that it doesn't have cycles. And as I just told you, in reality, it's a very hard to meet assumption, because usually when one thing influences the other, then you will also see reciprocal connection. And then, I'm sorry, nodes will represent random variables, as we saw, and edges between those nodes represent dependencies. And these dependencies are, again, causality is a separate thing. We will actually not address it too much. But the dependency is a simple fact that you will have a statistical dependence between the two variables. Each edge will be directed from a parent to a child. And then there's some value domains, and these domains usually are discrete. So you have a Boolean or some kind of categorical variables. But you could also have continuous ones. And I'll show you a few examples of that as well. So back to our confused students. Well, how does it look like? What we need to describe for those arrows is basically those conditional probability distributions. And I have two tables there. And those two tables are basically one way of explaining the data that we saw. And this way shows you basically that if you are paying attention, you have much higher chance of being confused, and not much, but a little bit higher chance of giving a correct answer. So what we want to do is we want to find the best Bayesian network given a data. And that's a kind of a plot that you see is taken from my domain, which is basically a lot of microarray slides lined up showing expression for different genes. And what we need to do is we need to have a scoring function that is giving us a mathematical notion of which networks are better than others. And we want to want those functions to be basically additive with respect to different variables. So we want to be able to compute it separately. So now this is tough. Unfortunately, the biggest problem so there's several ways of scoring those networks. I won't go into details. There's a lot of literature about that, but basically most of them are either based on Bayesian statistics or based on hypothesis testing. The bigger problem so there's two problems with finding those networks. One is that there is a lot of potential parent sets, but even a more difficult one is the fact that we need to make sure that there's no cycles in the result. And in order to deal with that, I would like to focus on cases where this is actually not an issue. So one such case is when you have a dynamic network. So for example, in cases where things are actually depending on of each other in time, so like imagine that you have the three different variables, like, for example, I don't know, finding a new technology, becoming proficient with this technology, then becoming bored with it, and then finding a new technology, and so on and so forth. So that's the kind of the situation that you might be familiar with. And then you have the actual network, resulting network of connections that has a cycle. And then what you can do is you can represent this with different sets of variables for a sort of before and after the change. And then with this kind of, so that's the bottom graph where you have basically the temporal relationship unrolled. And then you need to supply, for a training of such a network, you need to supply pairs of observations, so observation before and observation after. And that breaks the cycles and allows you to basically learn the structure that has a cycle if you then roll it back again. So that's the same network basically on both pictures. Another case that's quite similar, but it's even more important, is that sometimes you have different types of variables. So in this case, which is a little bit closer to home for me, you have mutations. They're a separate entity. You can have different pieces of parts of your genome mutated, or your patients can have it. And this can affect expression of different proteins. And again, there's many proteins, and you can have many of them, their expression changed. And then there's diseases, which again, you could be studying, for example, different subtypes of the same disease, or certain or different spectrum of diseases. And maybe in many cases, you would be interested only in certain types of connections. So for example, you may assume that you're interested in which mutations cause which changes, and which changes in expression cause different diseases, and not vice versa. We know that it can be vice versa. So for example, certain types of cancer can and do cause mutations, sorry. But you might just, for this purpose of building this particular model, you might not be interested in that. So that's kind of all about the introduction, the very basic stuff. And then what we developed a few years ago, and now we're further developing, is basically called a tool called BNFinder. It's a library. It's a Python library, open source. You can download it. And it works in this very case where you have a data set, a tabular data set like I showed you, that works under the assumption that cyclicity is satisfied by some external constraint. So basically, either it's a dynamic network, or you have some sort of constraint of the sort with this mutation and protein and disease case. It's relatively fast, and it actually finds the optimal solution. So how does it look like in an example? The simplest possible is basically we have two variables, P1 and P2, that are so-called regulators in this case. You can see it in the very top. This is the text file that is in the input. And you have this preamble with hash. There's a number of keywords that you can use. Regulators is just one. And then you have a tabular number of observations. In this case, it's inverted. So each column is actually an observation, and each row is a variable. And then there's a command line version of the tool. So BNF is basically shorthand for BNFinder, and you can specify that you want to use this file, and then it outputs the network that is visualized at the bottom. This is done with Cytoscape. So that's fairly easy. You can do this actually in parallel on number of cores. So that's very helpful, because when I said it's relatively efficient, well, for this particular situation where you have 6 variables, it really is very fast. But in cases where you have, let's say, dozens of variables and thousands of observations, it can already take hours on a single CPU. So then, since our computers now have multiple cores, you can basically use this -k keyword parameter, and then basically it gives relatively good efficiency. So as you can see on the graph, with our current implementation, we get about half of the linear speed up when we're using multiple cores. So that's useful. And what I can show you, what I will show you now is a few examples of a real-world applications where we used BNFinder in different settings. So the first case is actually, so that's mostly for the, you don't really have to get into details of that, but basically our collaborators found a way of measuring histone modifications or epigenetic modifications in different tissues. And what you get, the bottom part is the important part, you get these kind of data signals along the chromosomes. So this is your signal, and now they've measured that for a number of different modifications. So you have, let me count, 1, 2, 3, 7 different modifications, and they all run along a genome. That's a genome of actually fruit fly, which is a very nice model organism. And then you can see that these profiles are kind of rough. That's the usual situation with the experiment. And what we want to find is we want to find active regulatory elements. Whatever that is, what I can show you is here we have some prior knowledge from prior experiments. We have those blue and red and black bars. And you can see these are regulatory elements that are either active or inactive. And our point here is to first learn the dependence or conditional probability of a piece of DNA being active or inactive based on those profiles. So what we need to first find is to find we need to find a training data set. So a number of examples where we actually know what is the value of the variable that we want to predict. So luckily we had those collaborators who could have compiled this for us, and they've basically built this matrix of examples. So basically this is from pictures of embryos with staining. They have compiled this database, and basically this was our training set. So one important thing about this you might notice is that those profiles are continuous, or at least, well, we could discretize this signal, but it's not clear how many levels of signal you see there. Well, it's clear that there's some high levels, some low, and there's some different values in the middle. And it might be different actually for different signals. So one thing BNFinder can do is it can handle continuous data, which is unusual for a Bayesian network software. And what it basically does is, so here you can see an example of a network that uses this kind of data for explanatory purposes. So we have 3 variables that are regulating the 2 different outcomes, and they're all continuous. And in this case, both of the outputs actually depend on 2 different variables, and they depend in such a way that it's not easy to predict from one. So you can actually see on the bottom left part of this right plot, you see the green. I don't know how visible the colors are. You can see the green dots versus white dots, and this is our observations colored by the value of the x variable. And on the top right scatter plots, you see the values of y variable depending on the pairs of variables. So you can see that this basically, these are Gaussian variables, by the way. And in this case, what BNFinder does, it basically fits the best Gaussian distributions that it can do. It can find for those A, B, and C variables. So this is on the diagonal of this matrix. And subsequently, once those Gaussian normal distributions are fitted, it basically takes the posterior the probability of each of those observations to be in each of those observations to come from each of the distributions. So if you see at the very bottom, the C variable is a very clean bimodal distribution. So basically, you have two very distinct populations. So you have basically an underlying, so it's like a noisy generator. You basically have a discrete two states. And if you're in the lower states, like the yellow curve, basically, you have some variability, some noise in observation, but basically it's you can tell almost without an error that you're actually in the lower state. And same for the right. The red distribution basically is the excited high state, or whatever you want to call it, and then it's quite clear where you are. It's more interesting in the B or A cases where the distributions are kind of getting closer to each other. And there you actually are taking advantage of this probabilistic representation where you basically see that there is a middle ground. There's a place where the probability of an observation in this middle ground coming from either active or inactive state is about the same. But you can still see that this mixing is not uniform. So usually you can tell that you're, well, somewhere in the middle, but more like the active state or more like the inactive state. So this, basically the nice part of the modeling framework is that it can take advantage of this information. So I could discretize. I could basically slash my observations into lower and upper in this point where the red and yellow curves cross. But I can actually take advantage of this quantitative information, relative probability of coming from each of the distributions. So how it works, basically what we did for this activity example, this was used as a simple classifier in the end. You can see the ROC curve, and importantly for biologists, the model was actually quite intuitive to interpret. So you can see it on the right. There's 6 different variables that seem to be important. You can actually quantify this relative importance. You can actually assign positive or negative influence to each variable. And you can basically use cross-validation to assess the quality of this prediction. The model itself actually turns out to be the best model. Turns out to be only depend on 3 variables strong enough to include them, and it's non-additive. So it's important that this model could not be replaced by some kind of linear regression based model. Well, it provided some useful predictions, but I don't have much time, so I moved to another example. So that's how the input file would look like. There's a number. The preamble is much longer, so you actually have continuous. So you need to say which variables are continuous, which are the regulators, which can be perturbed, and then you can get. So that's actually a different data set that has a continuous and perturbed. So perturbed basically means that you can have things that are particularly useful for knockouts, but are basically any variable that was in a particular observation. You don't want to learn from this about this variable, then you can use this. A more complex example, just to get you even more confused, as this helps with comprehension, as we've learned in the beginning. That was a case where we built a bigger model where we had some missing data, and we actually used an expectation maximization framework. But the important idea is that you can basically construct the multiple training data sets with different values for your variable, and then iterate until you converge. And in this case, what I wanted to show you, just so that I actually do have a slide with actual code. So that's taken from the paper, from the supplementary materials where we have a code. There's a red frame. That's basically that's how you prepare the data. It's basically you can select from a matrix, and then there's a nice function that's called 2BNF, which basically generates a nice data set for well, basically file-like object that you can then use to create the proper data set. And then you have the function learn in a green frame. It's a little bit long for even this screen invocation of this function, but it basically has all the parameters that's needed. And then you have the blue, which is basically saving the conditional probability distribution with some parameters. And then for prediction, you do the data set very similarly, and then you use classify list, and then you get a list of classifications. So that's relatively simple, I'd say, and it also helped us to make these predictions of gene expression. So in summary, what I've told you is that these Bayesian networks is a useful, I would argue, mathematical model that can give you predictive models based on using conditional distribution probability distributions. It's open source, and it's relatively easy to apply to tabular data. It has a command line tool, and you can use it also as a library. It handles continuous data as well as the more usual discrete, and you can run it using multiprocessing Python package with relatively good efficiency, and there's some convenience function included, like cross-validation or ROC plots that's included, and there's a URL here, and that's not just me. I've been working on this for something like 5 years now with Norbert, who is my coworker. Most recently, Alina Frolova from Kiev was working on the new implementation of the parallelization, and she's trying. So we've been trying to make a cloud version of that, and I think we're close to that. Paweł and Agnieszka were involved earlier in making the second version of BNFinder that's most suitable for classification. And if there's any questions, I still have a few minutes to answer them. Thank you very much.
So I was wondering, what kind of questions are best sort of answered through this versus, all the other tools for machine learning that we have? So like, how does it work on high dimensional data sets, or is there some special use we should really care about this for?
So I would say that what we see from our experience, it's most effective when you have complex dependencies. So it can be used for classification, and it has competitive, let's say competitive performance, but it really shines when you have complex dependencies on not so many variables. Because if you have really hundreds of variables, then it's going to take long unless you have thousands of cores to find the model. It's also relatively good if you have multiple. So one nice thing that it works, for example, if you have those kind of, if your data is completely flat, so you have just hundreds of variables, each one of them the same, and you just want to kind of predict one other variable from them, then probably using random forests or SVMs is going to work for you very well. But if your data is hierarchical, so you have, let's say, 50 mutations, 100 genes, and then 3 different disease variants that you want to really predict from those, then you can use, the Bayesian networks will give you a benefit of actually using some of the hierarchy of your data. And it's also nice in the sense that in the end, it gives you these connections in an open sort of format. So in many ways, if you just use it for classification, and if your data is not particularly, the dependencies are not particularly complex, so if this is really if the variables, so for example, are independent, then why don't you use a naive Bayes classifier, because it makes the assumption of independence. So if it's satisfied, then probably you'll get the same thing with naive Bayes, for example.
Okay, and I have a question over here.
Yeah. Hi.
Using your library, how do you avoid not confusing correlation with causation?
Well, I'm not. I mean, it's it's not really possible to confuse. To kind of say, well, this is really causal. I mean, unless you do a perturbation experiment and you actually show that perturbing one, you're actually doing something to the else. It's not really excluding the possibility of the reverse still being true, right? So in a way, what we're saying, so there are other tools. So I'd say there's a few packages in R. There's Banjo that's written in Java. There's a number of tools that let you find Bayesian networks. And one thing we're doing here is kind of, instead of trying to fight this problem of finding a network that's acyclic, focus when this is really difficult computationally. We can focus on situations where we can make constraints. So for example, I do acknowledge maybe my disease is also causing mutations, for example, but I'm not going to try to find these. I assume that I'm only interested in mutations causing diseases, for example, right? And then using this framework can give me a very flexible tool where I can have very complex conditional dependencies in my model in a relatively easy to comprehend model. Because if I could use, for example, an SVM with some kind of complex kernel, but then it's a sort of a black box tool that's very difficult to reverse engineer basically what is this model actually using, for example, to make a prediction. And here, it's actually, I didn't really talk about this, but those scoring functions are usually taking into account some kind of sparsity of the model in one way or the other. So it's very different between them, how this is exactly achieved. But usually they're searching for a model that is not taking all of the variables, because then they're very prone to overfitting. I mean, you could make a Bayesian network from all of your data with all of the variables as parents of your single node that you're interested in, but then you'd most likely overfit to your data set. So those scoring functions will usually take this into account, and in the end, what you get is a relatively robust model, and it's kind of sparse usually.
Yeah, so you touched on continuous and discrete variables. How should we deal with sort of categorical variables? Can we do that within BNFinder?
Yeah. That's the default, actually. By default, it basically builds these kind of tables, kind of conditional probabilities, basically a frequency table. So like, for example, with this confused students or not. So you have those tables, and they grow naturally with the number of parents. They grow exponentially. So that's the only issue, but that's penalized in the scoring function usually. And there's also one important thing is that when we were developing this, we found that so usually when people prove when people discuss the difficulty of problem of finding the network, the optimal network, it's usually done under the assumption that you're dealing with the actual probability distribution, which is usually not the case. So usually you have your 1,000 or 10,000 or million observations, and those observations actually have a limited information content. So without getting into much detail of information content, this information content of your data is providing you with an actual fixed limit on the size of the network that can be best. So at some point you're reaching the threshold where you're actually adding too many parents to basically justify explaining the information in your data set. So actually, this is one nice thing that is rarely done when you're trying to find the best fitting model, is to basically limit. In this case, basically it's inside the scoring function to basically that there's a limit on how big the model can grow given the size of your input data.
So perhaps I'm mistaken, but it sounded like you said with even dozens or hundreds of features, it could take hours to find the optimal Bayesian network. And if that's the case, how do you handle input with millions of features? I mean, is there any way you can sort of downsample or aggregate those features, like maybe if it's labeled data, split it using a decision tree, and then at each decision level figure out how to merge multiple features, and then sort of have like compound features that you're then optimizing your Bayesian network on? Would that give you the results? Would that give you optimal results essentially?
Yeah, so I guess the short answer is that probably when you have millions of features, then most likely Bayesian networks, I mean, at least today, like in a vanilla flavor of Bayesian networks, is probably not the tool you want to use, because it's naturally you are considering a number of subsets of your millions of variables. So even if you have subsets of 2, that's quite a lot to look at. So I'd say that yes, most likely you could, if you have some way of, in an organized way, sub-sampling your features. I mean, I think that, yeah, probably you'd be better off using some other tools that are basically targeted, aimed at this kind of a situation. And then maybe you could, so I guess you could use something for feature selection, or some kind of removing the features that are clearly not related to the variable that you're most interested in explaining. And then use Bayesian networks to mine this limited set of features. So that's what I would do. So use something for like random forests to weed out those variables that are really not inputting anything, and then kind of have your few dozens of potentially important things to get the really complex conditional probability distribution from them.
All right, so let's give a round of applause to Bartek.